Hello and welcome everybody to my channel ominibioentrance, and today we will be discussing CSIR PYQs of the year 2018. I will be discussing the applied biology and methodology section. If you are new to my channel, please do like, share and subscribe. So coming to the next year, June 2018. Given below are few statements on agrobacterium mediated transformation of plants. Which one of the following statement is correct? tDNA transfer occurs from left border to right border. It occurs from right border to left border. So statement 1 is not correct. The GFP reporter gene can never be used for selection of transgenic plant. They can be used. So that is also not correct. Transformation frequencies will decrease on overexpression of virulence gene. That is also not correct. Host plant genes play an important role in influencing transformation frequencies. That is correct. So the answer here is option 4. Then next one. A 1257 base pair genomic DNA sequence of a prokaryotic gene was cloned under a strong constitutive promoter along with a suitable poly A signal and used for development of transgenic tobacco plants. So a 1257 base pair genomic DNA sequence of a prokaryote was cloned under a strong constitutive promoter and was used to develop transgenic tobacco plants. Molecular analysis revealed the presence of 3 types length of transgene derived mRNAs: 555 base pair, 981 base pair and 1257 base pair in the leaves of transgenic plant. So after transformation you got 555 base pair, 981 base pair, 1257 base pair mRNAs from the transgenic plant. The following statements were proposed to explain the above results. Which of the following combination of the above statements would correctly explain the obtained results? The 3 mRNAs represent alternatively spliced transcripts due to the presence of putative intronic sequence in the gene. That is correct. Option A is correct. The gene sequence was characterized by the presence of potential polyadenylation signals that resulted in premature termination of transcription. That is also correct. Expression of full-length transcripts, 1257 bases, was lethal to the transformed cells. If it was lethal, then you would not have got cells with 1257 base pair in the leaves of transgenic plants. But you got them, so it is not lethal. So that is not correct. The transgenic plants were chimeric in nature and comprised of a mix of transformed and untransformed cells. That is also not correct. So the answer here is option 3, A and B. Then next one. 3 students, PQR, in a research lab were trying to identify proteins that interact with the transcription factor X. P performed gel filtration experiments and identified that X was found along with protein A, B, C, D. Q performed co-immunoprecipitation experiment using antibodies to X and identified A, B, C. R did yeast 2 hybrid screen and identified only B. So 3 students, PQR, they were in the process of identifying protein that interact with the transcription factor X. So they performed gel filtration. One of them performed co-immunoprecipitation and another performed yeast 2 hybrid screen, and they identified and got 3 different results. Now the following are likely conclusions that may explain all the results. Which of the above conclusion best explain all the result? Now A, B, C, D are in complex with X. Yes, because as per gel filtration experiment, the X was found along with proteins A, B, C and D. So A, B, C, D are in complex with X. That is correct. X directly interacts with B. X directly interacts with B because as per yeast 2 hybrid system you identified only B. So answer 2 is also correct. Then next one, only A, B, C are in complex with X. No, D is also in complex with X. Then D is probably weakly associated with X. That could be the reason why you didn't get D upon co-immunoprecipitation with X. So answer is 1, 2 and 4. 1, 2 and 4 is option 2. Then next one. A researcher attempted to clone two genes X and Y independently in a plasmid vector for overexpression and purification in E. coli. All attempts to clone gene X were unsuccessful, whereas gene Y could be cloned easily. When the researcher attempted to clone gene X in the plasmid clone containing gene Y, gene X could be cloned. The following statements were proposed to explain the above results. Which of the following options represent a combination of correct statements to explain the observation? So, a researcher was attempting to clone two genes X and Y in a plasmid vector for overexpression and purification in E. coli. He was unsuccessful in cloning gene X alone, whereas gene Y he could clone easily. And now when the researcher attempted to clone gene X in the plasmid containing gene Y, gene X could be cloned easily. So what are the inferences that you draw from this experiment? That is what they have asked. Now, proteins encoded by gene Y is not lethal to the cell. That is correct. That is why you could clone Y easily. And not X. Then gene X has introns which prevents its expression in E. coli. That is also not correct. Then expression of X protein is lethal to the cell. That is correct. Sorry, B is not correct. Okay, expression of X protein is lethal to the cell. That is correct. That is why we could not clone gene X in E. coli. Then the Y gene product inhibits the activity of X protein. That is why gene X got cloned in the plasmid clone containing Y. So D is also correct. A, C, D are sorry, A, C, D is the answer. Answer is option 4. Then next one. Which one of the following set of essential components are required for Sanger method of DNA sequencing is in a required buffer containing MgCl2 and tris-HCl? So you will have DNA template, a primer, 4 deoxyribonucleotides, 4 labeled dideoxyribonucleotides, DNA polymerase. That itself is the answer. Answer is option 1. Then next one. The following cassette, this is from December 2018 question paper. The following cassette was designed to create estrogen receptor knockout mice. So SOH is site of homology. This is your neomycin resistance gene, and TK is thymidine light kinase. All these I have explained when I was explaining gene knockout. I don't remember the year, but you can refer to my previous videos for gene knockout experiment using homologous recombination methodology. Then, the following cassette was designed to create estrogen receptor knockout mice. What would ensure that proper recombination has taken place? Cells survive when cultured in presence of only G418. That cannot be, because they will survive even in ganciclovir, because they don't have the TK gene in them. Next one. Cells survive when cultured in presence of G418 followed by ganciclovir. That is correct. Cells die when cultured in presence of G418. No, they survive. Cells survive when cultured with G418 and die when cultured in ganciclovir. That is in the case of non-recombinance. So the answer here is option 2. The next one. Which one of the following statement regarding restriction modifying enzymes used in recombinant DNA technology is correct? Endonucleases remove nucleotides one at a time from the ends of a sequence. No, not from the ends but from within the sequence. Then type 2 class of restriction enzymes do not recognize palindromic sequences. They recognize palindromic sequences. Mung bean nuclease acts on double-stranded DNA or RNA termini. That is also not correct. Type 2 class of restriction enzyme can generate either sticky or blunt ends. That is correct. So answer here is option 4. Then next one. A DNA segment was cloned into the active site region of LAC-Z gene and the recombinant plasmid introduced into LAC-Z minus strain of E. coli and plated on a medium containing X-gal. The colonies showed blue color. Which one of the following statement is correct? So, a DNA segment was cloned into native site region of LAC-Z gene. So, when you clone something in the LAC-Z gene region itself, it will become LAC-Z minus and it will not produce functional beta-galactosidase enzyme. And this recombinant plasmid was introduced into LAC-Z minus strain of E. coli. So, the inserted gene is also creating LAC-Z minus condition, and E. coli chromosome also is having LAC-Z minus condition, and this was plated onto a medium containing X-gal. So blue colonies are formed. Now why are these blue colonies forming? So blue colonies are formed means beta-galactosidase is acting on X-gal, and it means somehow beta-galactosidase is synthesized. So we will see the options. The nature of the cloned DNA segment need not be special as cloning of any DNA in LAC-Z will result in disruption of its reading frame and production of blue color on X-gal plates. Now if there is disruption of LAC-Z, then blue colonies will not be formed. Then next option. The cloned DNA segment could be a group 1 intron whose removal from the precursor LAC-Z transcript in E. coli results in production of mature Z mRNA. That is also correct because group 1 introns have been identified in E. coli. So option 2 is maybe the right answer. The cloned sequence is likely to be LAC-Y sequence. That is also not correct. Then the cloned sequence is likely to be an anti-terminator sequence which allows full-length transcription of LAC-Z. That is also not correct. So the answer here is option 2. The next one. A researcher wanted to identify the enhancer sequence of a newly discovered gene. Shown below are the relevant regions of some of the reporter constructs the researcher designed to identify the enhancer. Which of the above constructs can be used to identify the enhancer? So first there is a promoter, then there is a reporter gene. In the a B condition, there is only reporter gene, which is not correct. Then there is SA and the reporter gene. That is also not correct. So only A is correct, where you have the promoter under and then the reporter gene. Next one. In a genetic assay, randomly generated fragments of yeast DNA were cloned into a bacterial plasmid containing gene X, essential for yeast viability on minimal media. The recombinant plasmid was used to transform a yeast strain deficient in recombination and lacking ISA-X gene. Transformants which survive on minimal media and form colonies should essentially have. Now, randomly generated fragments of yeast DNA, they were cloned into bacterial plasmid containing gene X, which was essential for viability of yeast on minimal media. The recombinant plasmid was used to transform. After that, the recombinant plasmid was isolated and it was used to transform a yeast strain deficient in recombination and lacking the X gene. So transformants which survive on minimal media and form colonies would essentially have. Now these recombinants, they are lacking, they don't have deficiency in recombination as well as lacking X gene. So they cannot grow in minimal media. But there were transformants which survive on minimal media and formed colonies. Now what could be the reason behind their formation of their growth on minimal media and formation of colonies? So yeast centromeric sequence which ensures integrity of the plasmid after transformation. That is not correct. Enhancers for essential gene missing in the transformed strain. That is also not correct. A sequence similar to bacterial origin of replication. No. Then yeast autonomous replicating sequence. Yes, because if ARS is present means they are capable of replication and can form colonies. So the answer here is option 4. Then next one. A Western blot analysis after treating cancer cells with prospective anti-cancer drug is shown below. So this is the without drug and this is the lane with drug. The following assumptions were made. Which one of the following combination is correct? Now CDK6 and the cyclin D1, they promote G1 phase. So the drug may have arrested the growth of cells at the G1 phase. That is not correct because CD6 and cyclin D1 increased in the presence of the drug. That is why you got a thicker band in the presence of the drug, and in the absence of the drug, the band was thin. The next one. The drug targeted the JAK-STAT signaling pathway. Yes, that could be correct because P-STAT and STAT3, it decreased in the presence of the drug. So that could be correct. B could be, option could be correct. You got only thinner bands with P-STAT3. The next one. The drug led to apoptosis of the cell. So there was cleaved PARP which is increasing. PARP increase means it is an indication of apoptosis. So drug induced led to the apoptosis of cell. That is correct because there was increase in the cleaved PARP upon treatment with drug. Then drug induced apoptosis was through extrinsic or mitochondrial independent pathway. That is also correct. So B, C, D are correct. Answer is option 4. Then next one. Given below are 4 statements related to agrobacterium mediated transfer of tDNA into plant cells. The correct sequence of events from earliest to the least is production of single-stranded tDNA by Vir D1 and D2 proteins, interaction of Vir E2 with VIP1 and Vir E3, use of Vir B, Vir D type 4 secretion system, activation of Vir A, Vir G complex. So the first stage in the sequence would be activation of Vir A, Vir G complex. That would be the first sequence. Then comes production of single-stranded tDNA by Vir D1 and D2 proteins. That comes second. Use of Vir B, Vir 4 type 4 secretion system. That comes the third. And interaction of Vir E2 with VIP1 and Vir E3. That comes fourth. So D, A, C, B. That is option 4. Next one. Construction of knockout mice may be performed using the Cre-Lox system. Eventually, the Cre recombinase of the bacteriophage P1 mediates site-specific recombination at a 34 base pair sequence. Logs P from the following statement. Choose the incorrect statement. So, this is a question from Cre-Lox P recombination system. So the first statement, the alteration of the chromosomal copy of the targeted gene requires a guide RNA. Now guide RNA, as you all have studied, it is involved in RNA editing. So first option itself is incorrect. It is not involved in Krylog system. So first option itself is incorrect statement. So the answer is option 1. Then next one. Given below is a table comprising various terms associated with plant tissue culture in column A and column B. Which one of the following options represent the most appropriate match between all the terms of column A and column B? So it is a match the following question. So auxin. Auxin is indole 3-acetic acid, A4. Protoplast culture. Protoplast culture requires pectinase and cellulase. So B3. Cytokinin is 6-furfuryl aminopurine. C2. And microspore culture is embryogenesis. D1. So the answer here is option 2. Then next one. Given below are names of techniques, column A, and their characteristic features or applications, column B. Which one of the following represents a correct match between column A and column B? So hybridoma technology. Hybridoma technology is for production of identical antibodies. A3. MALDI-TOF. MALDI-TOF is for determination of molecular weight of proteins and or peptides. Ion exchange chromatography, that is separation of protein based on their charge, and chromatin immunoprecipitation is for identification of protein complexes in cells. So answer here is option 2, A3, B4, C1 and D2. So next question. B3 proteins BLM1, BLM2, BLM3 were shown to be involved in repair of DNA double-stranded break. A chromatin immunoprecipitation experiment was performed for the 3 proteins. The pattern of results obtained is shown below. So 0 minute before the break. 0 minute before the break you have got the input, then 0 kb from the break, and this is the 1 kb from the break. Then you have got another table with 30 minutes after the break, and this is the input, then 0 kb from the break and 1 kb from the break. Now this is BLM1, BLM2 and BLM3. BLM1, as you can see, before the break BLM1 was barely synthesized. That is why you got very thin bands over here, whereas here you got a thick band, that is 30 minutes after the break. 0 kb from the break, you got a thick band, that means it was synthesized after the repair was induced and it went and bound to DNA. Similarly, not similarly, if you see BLM2 it is binding before the break. 0 minutes before the break it is binding, but once the break has occurred, BLM2 does not bind after the break. In case of 30 minutes after the break, you get only thin bands. Then BLM3, it doesn't bind in the beginning, but later it binds. So based on the above figure, choose the option that correctly interprets the data. BLM1, BLM2, BLM3 bind to DNA break sites. BLM2 does not bind after the break, it binds before the break. You can see from the table here, I have already explained that. BLM2 binds before the break and not after the break. If you see this tabular column. The next one, BLM1 binds to the break site. That is 0 kb from the break. It binds to the break site. That's correct. That is over here. You get a thick band over here. So BLM1 binds to the break site, or 0 kb from the break site it binds. So that's correct. BLM3 binds to the break site and beyond. That is why you got a band with where 1 kb from the break also BLM3 was found to bind. So it binds to the break site, that is 0 kb from the break, as well as and beyond, that is 1 kb from the break. So option 2 is correct. Now option 3, if you see, BLM2 remains bound to DNA after the break is induced. This is not correct because it gets dissociated after the break was induced. That is why you don't get bands over here in case of BLM2. BLM3 binds to DNA irrespective of the break. That is not correct. Before the break, there is no expression of BLM3. Only when the break has been induced, BLM3 binds to DNA at the break site and beyond the break site also. So option 2 is the correct answer over here.